colocar lá no YouTube, tranquilo? Ah, então tá bom. Depois a gente corta um pedacinho. Felipe, então vamos começar oficialmente, te agradecendo muito, né? Conhecer pessoas como você, né? Pesquisadores, pô, jovens que estão fazendo um trabalho incrível, é uma das grandes coisas que eu diria que eu ganhei no LinkedIn. Eu não tenho nenhuma rede social, entrei no LinkedIn há
Sim.
pouco, relativamente, né? Pouco tempo e estou adorando justamente por ter essa oportunidade, né? De conhecer profissionais como você que só pela literatura e tal seria muito mais difícil de identificar o nome, identificar também a pessoa, né? Numa rede social, você lê o perfil, você entende exatamente o que o cara gosta, o cara não gosta, você conhece a pessoa, é impressionante. Mas, Felipe, você vai apresentar esse seminário em inglês, né? Várias pessoas pediram para mim, e provavelmente colegas aí do exterior, deixa eu liberar aqui para fazer, e a gente vai receber, vai aumentando um pouco aí o número de pessoas entrando na sala ao longo do tempo, mas eu espero que não te incomode. Felipe, muito obrigado pela sua participação, como eu te pedi antes. Conta um pouquinho aí da sua história para a gente chegar saber como é que os Filipes são formados, né?
Tá ótimo.
Manda bala.
Bom, primeiramente, Gonçalo e também o pessoal do programa de pós-graduação aí, eu entrei em contato rapidamente com a Thaís. Obrigado, inicialmente, por me dar esse espaço aqui. Eu fico lisonjeado também por ter essa oportunidade, né? E honrado também de poder contar um pouquinho da minha história para os alunos, para os seus alunos e para os alunos de outros professores, outros profissionais. Né? Eu acho que é bastante importante a gente tentar manter a unidade dessa comunidade acadêmica que tem sofrido bastante ultimamente, né, nesses últimos anos. Então, é bem importante que a gente consiga manter essa dinâmica. Eu vou pedir para compartilhar a minha tela para eu tentar encontrar a minha apresentação aqui para a gente... Você, por favor, me informe se você consegue ver a minha tela.
Tá excelente.
Tá
Aí,
ótimo.
perfeito. Está em modo apresentação perfeito.
Okay, so I think I'll switch to English now, so we can actually start the presentation. Once again, thank you, Professor, for the opportunity. Please feel free to interrupt me during the presentation if my connection fails, for instance, or if there is some burning question that you need to do. So I'll start talking a little bit about part of the work that I published during my PhD, that we can give this fancy name of synthetic microbial communities for biofuels production, but it's a much more basic study that I have performed. But I will start covering with a summary where I will go through a bio, explain a little bit my professional history, then go to an introduction, and then I will deep dive with you guys on two hand-picked studies from my PhD. One specifically showing the strain dynamics of a specific contaminant bacteria and how they modulate the performance of ethanol biorefineries. And the one that has been recently published at Nature Communications, which dissects the interaction between these different species of bacteria and yeast in the industrial ethanol fermentation and how these interactions drive the process, with some concluding remarks afterward. So I'm a biologist by training. I graduated in 2008 at the University of São Paulo. And shortly after my graduation, I did my master's in the chemical engineering department. So I went to the engineering university, or in the engineering faculty, to be more specific, more precise, in the University of São Paulo. And during my master's, I worked with solid state fermentation and cellulase production for second generation ethanol. Shortly after my master's, I moved to the south of Brazil, to Curitiba, to start working at Novozymes, where I worked as a research associate and senior research associate. And my main focus there was not the only one, but the key focus there was to develop enzyme-based solutions for foam control in ethanol fermentations. So specifically in the case of the Brazilian biofuels fermentation, most of the foam is actually formed by the yeast. So many proteins in the surface of the yeast, since proteins have an amphiphilic nature, they have a hydrophobic and hydrophilic parts. They tend to make the yeast cells closer to the gas bubbles and therefore stabilizing foams. So we're able to develop an enzyme solution to remove these nanoproteins from the surface of the yeast and avoiding them to have this foaming phenotype. And after this project was closed, it's a product now in Brazil, it's being sold there. I decided to invest a little bit more in my formal education, and I decided to pursue a PhD. And since I had my bachelor's and master's in Brazil already, I felt the need to do that abroad. So I was fortunate enough to be accepted to a position in a PhD at the Novo Nordisk Foundation Center for Biosustainability. It's a research center located at the Technical University of Denmark, but it's almost completely funded by the Novo Nordisk Foundation, which is one of the leading funding agencies, private funding agencies in Europe, or also, I would suppose, in the globe for research. And there, the focus of my PhD was really to try to resolve the microbiome of sugarcane ethanol fermentations, and how can we translate these findings into microbial community interactions into more complex systems. After my PhD, I started a short postdoc as well in applied microbiology, where the focus was to develop novel foods via synthetic biology. So the idea was to reduce food waste by converting food that is edible, but is being wasted or lost during the production process into novel foods. So for instance, can you take potato husks or potato peels and make milk out of it? So that was the idea behind this project. And now I have recently joined AB InBev in the role of Global R&D Project Manager for Biotechnology and Bioingredients. So basically what I do here is that I develop the long-term strategy and coordinate the global research projects focusing on converting the side streams or the byproducts of breweries into high-value products using biotechnology. So the idea in the long term, in the long run, is to convert breweries into biorefineries. So that's basically the idea behind it. So going a little bit more into the introduction, why study microorganisms in the first place, right? So I guess it's mainly because they are everywhere. So from the soil to the surface of plant roots to the interior of the human gut, you can find microorganisms there, and they are seldom alone. Actually, what's common is to find them being part of complex communities. And these microbial communities, they play important roles in several natural processes, from biogeochemical cycles to the influence on human health, and also in biotechnological processes. And the functionality or the impact that these communities have in the environment around them and the composition of such communities are mainly driven by the ecological interactions of its components. So the species in a given community can have like a neutral interaction, or they can have a beneficial interaction, exemplified here as a mutualism, or they can have a negative interaction, as exemplified here as a mensalism. And it's important to stress that such interactions among species is context dependent. So two species might cooperate to degrade a complex biological sample, a complex biomass, but they might compete for the nutrients that are released upon its degradation, for instance. And what is the point of studying such communities and such interactions? By better understanding how microbial communities assemble and interact, one could, for instance, devise better medical treatments in the future, or improve the crop yields by modulating the microbiome of soil, or even developing novel or improved biological processes or industrial bioprocesses by using complex microbial mixtures or complex communities. But there are several pitfalls that limit the capacity to understand the dynamics of these communities at lab scale. Most often, it's difficult to reproduce environmental conditions precisely at laboratory scale, as well as it is to isolate species found in the environment. And also, there is the lack of defined and quantifiable parameters that one could use to try to understand the functionality of these given communities. So a simple model system to understand microbial interactions should be reproducible. So one could be able to reproduce that at lab scale. And obviously it should be simple enough that you could manage it to do that at lab scale. So you cannot have a lot of species. Also, you need some well-characterized species there. You cannot have some funky bug that you don't know how to grow or that requires a very specific nutrient that you cannot just add in the system. And clearly measurable parameters. So you need to understand what's the functionality that you're looking for and be able to measure and quantify that at laboratory scale. When we think about ethanol fermentations, they actually check all these boxes. So ethanol fermentation is a clearly well-defined biological process, probably the most well-characterized and studied biological process we know. It is highly reproducible, being even industrially applied. It's well-defined and measurable parameters. The ones that are most commonly known is yeast growth and ethanol production. And by itself is a relevant target for study. So I'm pretty sure most of you know that the sugarcane ethanol industry is a relevant industry for not only for Brazil, but globally. It produces several products out of fermentation and out of the processing of sugarcane biomass, and has the potential to produce much more products in the future. And it can play a leading role in fighting global warming if we play our cards right and actually are able to leverage this industry globally. Considering, again, the fight against global warming using this biotechnology, it is considered that the Brazilian ethanol has a benefit against gasoline on much lower CO2 emissions, about 90% less emissions than the relative CO2 emissions of gasoline. And in 13 years, it has been able to avoid the emission of more than 500 million tons of CO2 in the atmosphere, which translates to, for instance, planting 4 billion trees in the aforementioned period. So it's an extremely relevant industry and also has a socio-economic impact because it employs a lot of people. So by itself, it's a very relevant target for studying. And the ethanol in Brazil specifically is produced using a very peculiar fermentation system that uses very high cell density in fed batch fermentations. So these are very fast fermentations that operate under, as I mentioned, a very high biomass load. And after this fermentation is over, what happens is that the fermented beer is separated from yeast via centrifugation process. The beer is sent to distillation to recover for ethanol, and the yeast goes back to the process, but instead of going back to a new fermentation batch, it first is acid washed in a separate vat. And this is done every time before a new fermentation starts in order to first to keep bacterial population under a certain acceptable threshold, but also to deflocculate the yeast biomass. After this acid wash is done, the yeast biomass returns to the process and it starts all over again. And the mills operate like that throughout the entire crop season. So you actually have a stable microbiome over there that is being constantly fed. And you also have some resemblance of an immunological system here that is fighting off some unwanted contaminants. So you actually have a very stable microbial community to try to understand and study there, with a wealth of industrial data that is being gathered daily. And the efficiency of this process is partially defined by the interplay between the species found in this fermentation process. So both yeast and bacteria, they will compete for the substrate, for instance, for sucrose, and the metabolism of both species it will be inhibitory towards the other species. So ethanol is inhibitory for several bacterial species, as acetic acid is inhibitory to yeast. So this interplay between these different microorganisms will have a relevant impact on the performance of the industrial process. And contamination is still a major issue for this biofuels industry. Due to this sheer size of the process, it's not possible to work under aseptical conditions. So it runs non-aseptically, which leads to huge problems in contamination. So doing a very quick math, considering the average impact on contamination and the total ethanol produced by US and Brazil, the two major markets, one can estimate an economical impact of about $2 billion yearly on contamination alone. And besides the economical impact, there's also an environmental impact. The less ethanol you're able to make per amount of sugarcane, the more sugarcane you need to crush. So understanding the dynamics of this community is not only valid as a scientific endeavor, but also as an applied perspective to try to improve the dynamic of this process. Which leads us to the first study that I would like to present to you today, which is, describes how strain dynamics of specific contaminant bacteria modulate the performance of actual industrial ethanol biorefineries. This study has not yet been published in peer-reviewed journals, but I hope that will change shortly in the future. What we did in this study is that we sampled 2 independent biorefineries in the state of São Paulo throughout the year of 2017. We sampled 3 times each of these biorefineries, and we extracted the whole DNA of all the samples and sequenced it via shotgun metagenomics. After sequencing and using the proper bioinformatic tools, we are able to obtain a microbial community structure, which can be further compared with industrial process metadata in order to find potential correlations between community structure and process performance. After this, one can understand the gene pool or the total genetic makeup of this microbiome in order to identify key essential genes or pathways that can be deleterious to the process, as well as performing laboratory scale experiments in order to prove causality. So one thing is to find correlation, and the other is to actually demonstrate that at lab scale showing causality. And we are actually able to sample in all the unitary steps of the fermentation process. So we are able to sample in the broth, so in the fresh molasses broth, during the beginning, middle, and end of the fermentation, after centrifugation, and after acid wash. So we could have an overview on how the community is structured throughout all these steps and how these steps can actually be impacting the structure of such communities. And what we find here, and I do realize it's a very, very loaded slide, but please bear with me here. So the first thing we find that is extremely interesting is that independently from the mill, both mills have an extremely similar microbial community. So the mills are completely independent from each other. They are far away. They receive cane from different fields. But still, the top contaminating species from one mill are the same species from other mill. Also, another interesting aspect to understand and to follow is that the dynamics throughout the year. So if you see here on the bottom, the sampling time point, first, second, and third. So this is the first, second, and third sampling periods that we went to the sugarcane plant and sampled the process. The dynamics are extremely similar towards both industries. And finally, what is also extremely relevant is that the richness and the diversity of the community found in the broth for both mills is much richer and higher than the diversity found in the actual industrial process, which suggests that the stress imposed by the industrial process is such a strong selective force that this is the one that is actually driving the structure of this community. So you can throw a lot of different stuff here, you can throw a lot of different species, but there are only so few that can actually survive and thrive in this particular process. So this provides interesting insight that there seems to be a shared microbiome towards different ethanol plants in Brazil. That is really driven by the design of this particular process. And when we dive a little bit more and we try to understand the dynamics of different species, we also see that they share dynamics as well. So when we follow the fermentation of both of these meals from the beginning to the end, we see that for both cases, there seems to be a competition between Lactobacillus amylovorus and Fermento, which are the two top leading species on contamination in relative abundance terms. So considering the total number of cells, these are the ones that comprise the highest value of cells in the contaminating population. So for both meals, a similar dynamic is observed, where fermentum starts to take over part of the in the room inside the fermentation. Let's put it like this, when compared to Lactobacillus amylovorus. So now we actually have the dynamics of species, and we have the common structure throughout the entire year. So now we can actually start to make correlations with industrial metadata. The first thing we do is that using yearly metadata, we try to identify potential correlations between the process parameters itself. And we find almost a perfect correlation between ethanol yield and acidity. And these is actual industrial process parameters that the industry provides to us, which indicates that the higher the acidity, the lower the yield. And this is for both meals. The more organic acids you have in your system, the lower ethanol yield you get. The second obvious question you would ask is, who is involved in acidity? And we find that for the most relevant species, the one that's mostly strongly correlated with acidity, or actually the only one that is correlated with acidity, is Lactobacillus fermentum. Not only with acidity, but in bacterial cell counts, which tells you that the species that is actually growing in the process, the one that is actually increasing its relative abundance in terms of cell counts during the fermentation process, is most likely Lactobacillus fermentum. This guy is being favored in the fermentation process, but it's at the same time, it is harming the industrial process by increasing the acidity. And when we deep dive in the structure of the population of fermento, then here we are talking about strains. We are not even into species anymore. So we are able to have such a high resolution in our metagenomics analysis that we're able to identify different strain clusters of Lactobacillus fermentum. And we also find similar patterns between the two meals. So depending on the cluster that dominates the population of Lactobacillus fermentum, the performance of the fermentation can be either low or high. So if we have one specific strain cluster, which is this dark red called 2, that actually seems to be beneficial or less detrimental to the fermentation process, whereas there are 2 other strain clusters that seems to be extremely detrimental to the process. So strain cluster 1 and 3. And actually, we can even identify the fate of the fermentations in the acid wash. So depending of the strain cluster that is found during the acid wash of industrial ethanol fermentations, we can correlate that with the performance of the fermentation in that given day, which suggests again that it really doesn't matter the diversity that is coming into the molasses. What matters are the species that are actually able to thrive in the process and survive. But what drives this difference? What drives this shift among different strain clusters during the acid wash, these particular strain clusters of Lactobacillus fermentum? It's temperature, actually. So the higher the temperature during the fermentation on that given day, the higher the relative abundance of detrimental species or detrimental strain clusters of fermentum in the acid wash. We can even observe that at lab scale. So higher temperatures benefit Lactobacillus fermentum when compared to amylovorous. That's a given. We know that from several studies. What we show here is that higher temperatures benefit differently different strains of fermentum. So the increase in growth rate differs from strain to strain. And this difference in growth rate might explain why there is a dominance of given strains, and more specifically detrimental strains when temperature goes higher. we also prove the causality of this phenomenon in controlled laboratory fermentations. So when comparing the yield of the fermentation with standalone yeast fermentations, in the presence of specific contaminant species and isolates from the industrial ethanol fermentation process, we find that the only one that was able to cause a significant drop in yield was a specific strain of Lactobacillus fermentum. And when we observe the metabolic profile or the metabolite profile of this strain, we also find that there's a key difference among other strains, suggesting that indeed acidity is the driving force of the reduction in yield, and this is indeed strain-driven, corroborating the findings that we observed with our metagenomic and bioinformatics study. And we also find interesting, at least in these conditions, that some strains or some species are actually beneficial to the process, which leads us to our second study, which is the complex yeast-bacteria interactions and how they affect the yield of industrial ethanol fermentations, this study has been recently published at Nature Communications. In order to understand that, we rationally dissected all the interactions found in this microbiome. And how we did that is that I performed several parallel fermentations with all the possible combinations of yeast and bacteria found in this process by using the top 6 most common strains and species in this process, which comprises in relative abundance terms about 80% of the diversity in this community. So if there's an impact there, most likely it's coming from these guys because they just dominate the microbial community in relative abundance terms. And after these fermentations were completed, the composition of the community was analyzed via flow cytometry in order to for us to understand the relative abundance and the total cell count between yeast and bacteria. And the main metabolites were analyzed and quantified via liquid chromatography. The first interesting finding that we observed is that the yeast population is actually favored by higher complexity communities. So the more species you have in the system, the more bacterial species you have in the system, the higher is the relative abundance of yeast in the total population or in the total microbial community. And this happens because the more species of bacteria you add, the lower is the cell count of the bacteria. And which suggests and indicates that these bacterial species are actually competing for substrates and for nutrients that they are not competing or not sharing with yeast. So yeast is impervious to this particular competition. So you can imagine, for instance, lactobacilli are extremely fastidious organisms, so they will be competing for certain amino acids. And on the other hand, yeast is able to produce all the amino acids it needs. So they're most likely competition for specific nutrients that yeast is not being affected for. And we also try to understand and to create a mathematical model that could predict the dynamics of this microbial community. And we did so by creating a logarithmic equation which correlates, which is a linear correlation between yeast cell counts and ethanol production. So the higher the ethanol, the higher the functionality. That's the final goal. So the higher the ethanol, the higher the functional level of the community. And we can understand this functionality. We can break down in smaller parts by adding, for instance, the pairwise interactions, which is basically the functional in the presence or absence of specific species, and the residual interactions or the residual effect of the functionalities, which we call higher order. So as a simple explanation, higher order interactions are interactions that are found in communities with 3 or more species, and how the presence of a given species changes the interaction between 2 other species. So 2 species might compete against each other when they are growing alone, but when you add a 3rd species, the interaction among these 2 species change. So that's basically how higher order interactions are explained. So when we when we measure all the functionality of all these fermentations we performed, we have a map, a functionality map or a functional landscape of how this community is structured and how it behaves based on the number of species. So this is the observed functionality, which is somewhat stable throughout the process. But when we create a simple null model comparing only the presence or absence of species, this does not precisely explain what we were able to observe. So we are not only talking about presence or absence of species. It's not an additive model. So we considered adding, as I mentioned before, the pairwise interaction levels. Turns out that most pairwise interactions, so when you have two species growing together with yeast, most of the interactions are negative. So a bacterial species growing alone with yeast seems to be behaving better towards yeast when it's grown with in the presence of another species. When we calculate, when we estimate or create a model considering the pairwise interactions, we have actually a much worse model that does not explain reality either. So pairwise interactions are not the forces that are actually explaining what we are observing in the process. Indeed, what we find is that higher order interactions buffer out these negative pairwise interactions. So the more species you add in the system, the more diluted these negative pairwise interactions become. So the more diverse the microbial community is, the more stable the process runs. So in the end of the day, that's what this mathematical model is telling. And when we analyze the structure of all these fermentations, we find that one specific species that I have also explained in the previous presentation, in the previous study, actually seems to benefit the yeast population. So whenever the community composition structure is, when you have Lactobacillus amylovirus inside it, you have higher ethanol yields and you have higher yeast cell counts. So why is that? In order to understand this, we created metabolic models from both yeast and bacteria and performed flux balance analysis to try to understand what is the interaction happening between these two species. The model predicts that acetaldehyde is produced from the metabolism of Lactobacillus amylovirus, and yeast cannot take that as an electron acceptor. So this is what the simulation in silico is suggesting to us. In order to validate that, we grow yeast in the presence of different concentrations of acetaldehyde, and indicates that in higher concentrations of acetaldehyde, there is a benefit for growth rate and also ethanol yield. This leads us to try to understand a model that acetaldehyde might work as an electron acceptor by providing more substrate for yeast to convert NADH to oxidize NADH into ethanol, resulting into final ethanol. And when yeast is growing, it also converts some of the carbon source into glycerol because it needs to balance its cofactor pool. But if you have more acetaldehyde in the system, yeast would not need to produce such a high amount of glycerol, thus resulting in a higher ethanol yield and a higher pyruvate pool that could be converted into higher biomass. So if this hypothesis, if this model is correct, the higher the availability of acetaldehyde in the system, the lower is your glycerol titer. And this is exactly what we find in these particular fermentations. So the higher the amount of acetaldehyde in the system, the lower is the glycerol titer produced by the yeast, suggesting that this model is probably not wrong. So moving on to the concluding remarks, we are able to break down into fundamental and applied conclusions of this study. And we see and we show and we prove with with experimental data that strain level variations are enough to influence a community's functionality. So is a higher level of complexity. We're not talking about species anymore, we're talking about strains. And sugarcane ethanol fermentations are actually a very good model to try to understand microbial community dynamics. It's very easy to reproduce at lab scale. They are very stable, and you have a wealth of data that you can easily quantify. High order interactions, just like it's found in other microbial communities, seems to play a key role there also on stabilizing this community against negative pairwise interactions. And the cross-feeding between species seems to be a key ecological driver of this community. So there is a key species there, like the Bacillus amylovirus, that seems to be benefiting yeast. At the same time, there seems to be detrimental strains of fermentum that are impairing yeast fermentation by the organic acids being produced. So we find that in a fundamental set of conclusions, it is critical to consider higher resolution when doing metagenomic studies in microbiome. Also, in the applied conclusions, we identify specific strains of fermentum that were able to reduce the fermentation yield in almost 4%. If you convert that into gains, into value, it's about $900 million per year of money that is being lost by ethanol that is not being produced because this guy decided to make a lot of organic acids during the fermentation. And the ecology of the microbial community is actually critical for the process to run. We have been considering this ethanol production process as a sole or unique fermentation of yeast, but this is not the case. What you have there is that you have yeast and a lot of other species that are fighting for nutrients and room. And the idea is to better tailor these communities into a structure that actually makes sense to the process. So instead of for instance, of considering using broad range antibiotics or trying to reduce the population of the bacterial community as its minimum, the most rational approach, as it seems, as the data suggests, is to use a selective approach that was used with yeast back in the 90s when we started selecting for the best industrial yeast strains. Maybe it's time to start selecting for the best industrial bacterial strains as well. So there are some strains you don't want in the process, and there are some strains that you actually need in the process. So this is also the key takeaway message from this particular applied conclusions. So there are beneficial bacteria, and there are detrimental strains that needs to get removed selectively in the process. Finally, I would like to thank everybody that was involved in this study, and my colleagues from the Novo Nordisk Foundation for Biosustainability, my collaborator at the University of São Paulo as well, Professor Thiago Basso, my collaborator from Hans Knoll Institute, Gianni Paneggiato, and the professor that hosted me at Yale during my study to try to actually create mathematical models and in vitro simulation from flux balance analysis of this community, Professor Álvaro Sanchez, and the industrial partners, Gilor, San Martino, that were kind enough to provide us not only access to the industrial facility, but also access to industrial metadata, and the financial support of the Novo Nordisk Foundation. And I finish leaving the contact of my former supervisor, Professor Martin Sommer, and myself, if you would like to connect in the future and ask some questions. More than happy to answer if I am able to do so. And yes, that was it from my side. I hope you guys have enjoyed, and I hope it was not too much in a hurry. I have been having meetings the whole day, so I may be a little bit too fast, but I hope I was able to make sense.
Felipe, it was a really great meeting, really great presentation, wonderful work. The people that is watching, please, questions are open, and while they formulate the questions, I'm understanding that you have a process that you probably are going to patent. I don't know how much you can speak about this, but the obvious conclusion of your excellent work because everybody knows about the role of the bacteria in the fermentation, but always thinking in the negative way. That means the bacteria are there to compete with the yeast. However, we have seen in some occasions, but very, very difficult to systematize and to find a clear conclusion, that in some cases the presence of bacteria could maybe be helpful, but never in such a way that we could really get a conclusion as you got now. Really great work. And my question is, I don't know if you can answer this, but do you believe that you could really replace antibiotics using a given proportion of bacteria, that the bacteria, as you are showing, is able to stimulate a probably more complex community that is not bad for the yeast? On the contrary, could be good for the yeast.
Mhm.
And I do believe that you are going probably to patent something or to establish a process to do that.
Yes, there are ongoing discussions with a Danish startup that has a technological tool that is able to selectively target specific strains of bacteria in a process, and they are willing to move more towards this application. I cannot disclose much more than that, but answering your question, I do believe it's possible. It's certainly not impossible to replace antibiotics for biological solutions. I certainly believe so. And I guess the real trick there, as we were able to show in the metagenomics study that we performed, is to find strains of, for instance, Lactobacillus amylovirus or a mix of other strains that are actually able to fight back the growth of Lactobacillus fermentum during fermentation in higher temperatures. So amylovirus is more sensitive to fermentation when compared to fermentum, and that's why it loses room during the fermentation process. But we also show with actual industrial metadata that the higher the amylovirus proportion in the relative abundance of the community, the higher the yields. So it is really a matter of trying to understand what is the impact there is. Sometimes like a chicken and egg problem, like what is causing the drop in yields? Is the higher temperature, or is the fermental population that is growing? Data tends to suggest that it's fermentum population that is growing. Some strains produce lots of organic acids. These organic acids are inhibitory to yeast, and therefore you have a reduction in yield. So fighting off these particular strains, so shaping is not even shaping the community, it's shaping a specific population. You can have fermentum there, but as long as you have the good guys. So if you're able to find a specific bacteria that can remove this specific strain of fermentum, that would provide probably a gain.
You know, in my opinion, you have shown something, a really new strategy, because this is a problem in first generation, but in second generation, this is much more sensitive. I have seen several times second generation fermentation that just stopped full of sugar because organic acid levels was so high that stopped everything. And what you are saying, Felipe, in your first approach, I would say, because what you are doing now is opening a completely new area. I could, for example, take a bacteria like this that you have found is important and make a specific bacteria. So genetic engineering or something special for designed for this goal that you have shown that is possible and is beneficial. That means you are opening a number of possibilities that we can exploit in order to clearly place antibiotics. I do believe that antibiotics in the future, in the near future, will be forbidden. We are always in the fermentation, probably because we have no other option. But I don't see this as a sustainable, let's say.
No, not at all.
Yes.
Not at all.
Felipe,
I totally agree.
yeah. You know, for that reason, I wanted you to be here because I can recognize when I see something out of the box that can really have a big future in the sector. And this is one of these findings. I have no doubt about that.
Yeah, I guess it's a matter of not thinking about, sorry to interrupt, Professor, but I guess it's a matter of stop thinking about species and thinking about metabolic potentials.
Perfect.
So you need a certain genetic makeup in the process to get the most out of it. So it's not only replacing antibiotics, but increasing the yield by having complementary metabolic profiles. So this bacteria, for instance, amylovirus, as the name tells, it's able to degrade starch. And it is producing a pool of acetaldehyde that yeast is able to consume. So what it's actually doing is that part of the carbon that was not accessible to yeast before, now it is in the form of acetaldehyde. And that's why it leads to higher ethanol yields, even when it's growing in co-culture, even when you have only amylovirus and Saccharomyces cerevisiae without the presence of fermentum, you have higher yields. And this is why there's a complementary metabolic makeup there. And this goes beyond the replacing antibiotics. This goes actually on modulating the metabolic system that you have there to get the most out of the carbon that you're adding. And I guess many industries now are considering having complex communities growing because they actually grow better. So you actually have a more efficient system when you have different species growing together.
Totally agree with you. Totally agree with you, including when you have bacteria that are expressing amylase, you see that the fermentation efficiency is higher. You have more. That means you have bacteria that you don't know exactly which kind of enzyme specifically is working, but you have a lot. You have a big number producing something that can be directly utilized by yeast. It's great. I totally agree. These two phases. But again, I think that if we could treat the fermentation tanks with some bacteria like this, avoiding antibiotics.
Yeah, I guess this solution would go hand by hand, right? Because it's also in the system, especially in the Brazilian system with cell recycling, it's very hard for you to keep, for instance, GMO strains of yeast there throughout the process because they tend to get washed out by more robust strains.
Yes.
So why not dividing the labor and allowing the bacteria to do part of what the GMO yeast would be able to do, for instance?
Yes, perfect. And you know, look, if you had the possibility to have a GMO bacteria playing a role in improving the fermentation quality, in this case, you would not have the problem of washing out the microorganisms because you could put the microorganisms at each new round because the concentration is very small and this is something really as a nucleus. But Felipe, Jaime has a question. Jaime, please. I think that Erwin Franiak has also raised a question. I don't know, Erwin, if Felipe already answered with the presentation. Possibly yes, but please. Jaime, please.
Okay. Good afternoon, everyone. I am very happy to see my friend Felipe and my friend Gonzalo discussing something that is very important to me. I am very happy to see how much we are aligned in the concepts that Felipe brought to us today. Very, very important. Just to give an example, for many years, many people thought about sterilizing the must. They thought about sterility in the full plant. That would be impossible and very expensive. What Felipe is showing us is that most likely we don't need to think about this sterility points. So also, I agree with you that the acid wash is not only to treat the yeast, but is to select the bacterial population. That is very, very funny, very important. And for many years, I've been talking about this, but people is still thinking on sterility, on antibiotics, biocides, and putting a lot of sulfuric acid to dissolve the plant in order to fight bacteria. So my only question today, of course, I would like to talk hours about this, but the question I have today to Felipe is the following. Is it possible to develop a test kit, a simple test, in order to detect the good guys and the bad guys? We just could take one drop of the fermenting must, putting on a plate or something like this, and in minutes, we could know if, let's say, the good guys are there or the bad guys are there, and look for solutions on this. This is my question today. Thank you very much, Felipe.
Thank you, John. Thank you for your presence here and for the question. And yeah, I certainly think it's possible, because I mean there are enough genetic differences to cluster these negative strains in specific strain clusters. So one could devise, for instance, targeted genetic tests or genetic kits with to amplify specific regions of the DNA of this particular species in order to correlate them with the relative abundance of the total species. I mean, you even have nanodrop today that can do that really fast, but one could think in a more targeted method using specific primers that amplify unique regions of these particular strains. And then you could have your answer, for instance, in a few hours or so. How contaminated is your process in the bad guys?
Good.
And the same goes to the good guys.
So very nice presentations. For me, always new. I'm not involved on processing of ethanol, but I learn a lot of in your presentation. I think very deeply and didactical form to understand what happens. But one point that's for me not very clear in page 10, when you present a lot of evolution in 3 steps, in 2 meals, different meals, and we see that you have one case, the fermentous bacteria is the keep it there, and another is this I don't know the name, this blue one, this amorphous bacteria is more resistant to the meal B. And then my question is, during these steps, you have evolution of all quantity of bacteria, and these have a bigger evolution in quantitative analysis, not only in percentage. I don't understand what happens with another one. Is destroyed, the another bacteria, or is not only evolved on this process, and another grow more quickly? Only to understand what is the tendencies to destroy or to grow quickly, differently regarding the different of meals and different of process.
I guess you mean this slide.
No,
right?
This is 20, page 20, 21. Yes, exactly.
Yeah. So what I believe is happening, and we need to also take in consideration the design of the process. There's a fed batch system, so the volume is increasing. So the volume of the fermentation is growing throughout the fermentation process. And we have seen already, I have shown you guys that what stays in the process, the bacteria that grow are actually the bacteria that starts the process. So what's happening is that this guy is growing faster than this other guy. And this is relative abundance terms. So how much of the species or the total number of bacterial cell counts does this species answers for, and how much of the relative abundance this species answer for? So not necessarily they are being destroyed, but they are being diluted by this one that is growing more.
Okay.
So this is probably what's happening, considering the structure of the process.
Okay, thank you. Very, very interesting. Thank you very much.
Thank you.
Erwin, you pointed something very interesting that is not normal when people think in fermentation in small volume. Actually, large part of the fermentation occurs during the feeding of the tank. Large part, that means you have fermentation from beginning to the end, and not completely, but very different because of the volume that is inside. It's having ethanol fermentation in Brazil. This is one of the more complex ecological interaction that you have, I would say, in the industry, no question. It's the most complex. But I would say, as a biological phenomenon, that Felipe is helping us really a lot to understand, to think about not only the science, but also how to utilize the science to make technology and real innovation. And it's very good that you are doing this so nice scientific approach together with companies interested in solving problems. This is my opinion.
Yeah. And this is something that driven me also to go abroad, because it's pretty clear that we cannot answer the problems we have today by only replicating the tools and the methodologies we have been doing for years. So we need to bring new stuff to the table. So I was fortunate enough to be accepted in this lab that has a deep expertise in human microbiome studies. So they have a very strong expertise on metagenomics and bioinformatics. And on the other hand, I have quite some background in industrial biotechnology. So I could bring my expertise on ethanol fermentation and use their expertise on microbiome studies and microbial ecology to try to correlate both. Because that's where I feel we still have a lot of gap. Obviously there are some very nice studies coming out, but there is still quite some gap in Brazil when we see most studies are still using metabar coding. Metabar coding goes up to genus level, and we see that the devil is in the detail. So the difference is in the strain level, not in the genus level. So you need more firepower to try to understand what's going on there.
Totally agree, Felipe. Totally agree. Bom, vou passar agora para o português para me despedir aí de todo mundo. Agradecer Felipe novamente. Excelente seminário, Felipe. Semana que vem, pessoal, nós teremos mais levedura. O Wesley Marques, que foi, acho que o Felipe deve conhecer.
Conheço.
Isso, exatamente. Eu aproveitei para colocar vocês dois, assim, para a gente ter uma sessão, um aprofundamento em levedura, que é quem faz o trabalho, né, de converter alguma coisa sólida no líquido, que é o que foi o que fez o grande sucesso dos combustíveis fósseis, né? Então, o etanol tem essa característica extraordinária de ser líquida, com grande densidade de energia, e quem faz isso são as nossas amigas leveduras, de forma extremamente generosa, utilizando uma quantidade mínima, né, de energia para converter uma coisa na outra. Felipe, te agradeço demais, despeço aí de você, de todos os amigos aí que acompanharam, está no nosso canal no YouTube, de forma que eu acho que essa sua aula tem que ser consultada por todos e por muito tempo.
Gonçalo, eu
um abraço, Felipe, mais sucesso, e é isso aí. E agora
contato com vocês.
Gonçalo.
Né? É isso aí, estamos juntos.
Oi, Gonçalo.
Opa,
quem está
amigo.
falando com você é o professor Mário César, tudo bem?
Ô, Mário, tudo ótimo.
Eu estou fazendo o meu doutorado aí com vocês, tá? E eu tenho uma experiência em controle microbiológico de processo de fermentação há mais de 20 anos. A minha pesquisa junto com a Unesp, que é de Araraquara, é com radicais livres para uma desinfecção do século XXI. Nós já finalizamos, já tem usina usando e com uma performance exatamente interessantíssima a nível de produção de etanol, chegando a produzir por volta de 1, 2, 3% a mais de um total de processo de fermentação. O que me traz assim uma pergunta para o nosso colega, que acabou não dando tempo, é que existe um problema grave em termos de sustentabilidade o uso inadequado de antibiótico dentro do nosso setor. E isso está trazendo, antes de mais nada, bactérias extremamente multiresistentes, acabando com o solo, e, além disso, devido à condição que essas bactérias ganham no meio ambiente de se relacionarem com outros gêneros e espécies de bactérias, através de regiões plasmidiais, passando exatamente esses genes de resistência. Então, eu gostaria que vocês ficassem atentos, sabe? É um convite, inclusive, de uma forma muito humilde, vocês ficarem atentos a esse trabalho que está sendo realizado de radicais livres aí. Eu agradeço aí pela posição, pela deixa de vocês aí. Muito obrigado, Gonçalo. Você já me conhece há muito tempo. Nós conversamos isso num summit em São Paulo, isso acho que há 7 ou 8 anos atrás. E eu dei continuidade e eu consegui finalizar o trabalho. Esse é o meu segundo doutorado.
Se puder me mandar mais informações, até pelo LinkedIn, é uma informação relevante para a gente distribuir.
Pelo LinkedIn, acho que você já leu alguma coisa minha.
Eu acho que sim. Bom,
eu te agradeço.
Agradeço, amigo, agradeço mesmo. Acho que é um tema de enorme importância para a indústria brasileira, para a indústria de etanol. Então, novamente, pessoal, despeço a todos. Felipe, novamente agradecendo a todos. Aí, o Jaime botou até um coração aí para a sua apresentação, né? É isso aí. Pessoal, boa tarde a todos, viu?
Muito obrigado, viu? Um bom dia para vocês.
Um abração.
Tchau.
Excelente, Felipe. Muito obrigado.